Mendel discovered a mode of inheritance called complete dominance. This is a genetic phenomenon in which one gene controls a trait. There are two alleles per gene. One allele is dominant and the other is recessive, and the biological sex of the offspring is irrelevant with respect to the trait. Other modes of inheritance have since been discovered. If there's a situation in which more than one gene controls a trait, that is termed polygenic. If there are more than two alleles per gene and two of them share dominance, that is called codominance. If there is no dominant allele but three phenotypes in which the heterozygous appears intermediate, this is called incomplete dominance. If a trait is determined by the environment and not genetics, this is called environmental influence. Finally, a trait in which one sex is affected more than the other is likely to be carried by the sex chromosome, and therefore this is called an X-linked mode of inheritance. So let's get started with incomplete dominance. In Mendel's experiments, he concluded that there were two alleles and that one allele was dominant to the other. However, we now know that in some cases, a behavior similar to blending inheritance occurs, which we now call incomplete dominance. This happens when there is an intermediate phenotype made from the breeding of two parents with different phenotypes. The best example of this is snapdragons. Snapdragons are one of the earliest studied examples. So in this case, a red and white snapdragon make 100% pink offspring, leading us to conclude that the heterozygous offspring are some form of intermediary. When two pink snapdragons are bred together, the result is a 1:2 to 1 red-pink-white snapdragon ratio. Mendel also concluded that there are only two alleles per trait and that one of them is dominant to the other. We now know that two alleles can be equally dominant to each other, and when both alleles are inherited, both phenotypes are displayed. Human blood type is a great example of codominance, as there are four types, now not including the plus or minus rhesus factor, of course. The rhesus factor, which determines whether you have positive or negative blood, actually follows Mendel's rules of complete dominance. But the A, B, AB, and O blood types do not. So blood type is based off of a certain blood surface glycoproteins. A and B blood types have their respective markers, while O blood has none. There are four blood types, and here are their genotypes. I represents dominant, but must contain the superscript A or B since both are equally dominant. O blood is designated by two lowercase i's since it is the recessive blood type. Therefore, A and B blood types can carry an O allele. AB blood type has both an A and a B allele, so both cell surface glycoproteins are displayed. Let's do an example problem. So what is the genotypic and phenotypic ratios of the offspring between a man heterozygous for B blood and a woman with O blood? So if a man is heterozygous for B blood, his genotype will be capital I, B, lowercase I, and then the woman's will be two lowercase i's. And that ends up with a genotypic and phenotypic ratio of 50-50. And then the phenotypic ratio would be 50% of the time you would expect a child with B blood, 50% of the time you would expect a child to have O blood. So what that means is that child 1 has a 50% chance of having B blood or 50% chance of O. The second child has the same percentage, so does the third, so does the fourth. It is quite possible all their children could be type B or all their children could be type O. Those probabilities reset with every birth. In reality, there are three cell surface glycoproteins, A, B, and D. The D glycoprotein is known as your rhesus factor and determines if you are positive or negative. The presence of the D allele indicates a positive rhesus factor, while negative means there is no rhesus factor or no D allele. So your blood type may be A positive, but your genotype should be one of the four displayed. So here's some interesting facts about blood donation. First, your blood type must be matched to an appropriate type, or your immune system will attack the foreign blood and cause a life-threatening blood clot called hemagglutination. The blood cell surface molecules are identified by antibodies that will attack the opposite. However, most people can receive more than one blood type. O-negative blood can be received by anyone. That's because O-negative blood has no cell surface recognition molecules for antibodies to attack. They lack the A, B, and D cell surface molecules. Unfortunately, O-negative people can only receive O-negative blood. This is why it's especially important for O-negative people to donate blood if they can. AB positive blood contains all three cell surface molecules, A, B, and D, so they can receive any blood type. In Mendel's experiments, he concluded that only two alleles, one dominant and one recessive, existed for his pea plant traits. We now know that while a single individual can only inherit two alleles, a population may have more than two alleles for a given trait. When many alleles for a given gene exist, the most common phenotype or genotype is often called the wild type, or sometimes it's just abbreviated with the plus sign. All other existing phenotypes and genotypes are considered variants that deviate from the wild type. In a certain rabbit species, there exists four alleles. Capital C for full color, which is the wild type. There's C, a lowercase c with a superscript ch for chinchilla, which is described as a black-tipped white fur. So lowercase c with a superscript h for Himalayan, which is described as a white fur with black paws, nose, ears, and tail. And finally, just lowercase c, which refers to an albino or a white-furred rabbit. Provided that the capital C allele exists in a genotype, the rabbit will have full colored fur, but will carry any of the other fur varieties. Chinchilla varieties happen when a rabbit inherits a lowercase c superscript ch, lowercase c superscript ch genotype, a lowercase c superscript ch lowercase c h genotype, or lowercase c ch lowercase c genotype. Chinchillas are more dominant than the next two fur types. Himalayans happen when a rabbit inherits a lowercase c superscript h lowercase c superscript h or lowercase c superscript h c genotype, and in albinos are the result of a homozygous recessive genotype with two lowercase c's. So let's say we breed a heterozygous chinchilla-colored rabbit with a heterozygous Himalayan rabbit. When you go to break these down, these are the following genotypes that you will receive. Your genotypic ratio would be a 1-to-1 to 1-to-1 ratio for all four genotypes. However, there's only going to be a 50%, 50%, or 1-to-1 ratio of chinchilla to Himalayan fur types. Mendel also concluded that all traits in his pea plants were genetically determined, but we now know that is not always the case. Most phenotypes are strongly influenced by the physical environment in addition to their corresponding genotypes. For example, while human height is genetically determined, the lack of nutrition could lead to stunted growth. The combined effects of gene and environment is referred to as gene by environment interaction. So while you may inherit the potential for building larger muscle mass, your environment is needed for you to achieve these goals. In hydrangeas, the soil pH determines the petal color. Pink petals are the result of more acidic soils, while blue is the result of more alkaline soils. It's like having your own litmus test in a garden. Early in the 20th century, Thomas Hunt Morgan adopted fruit flies, also known as the scientific name Drosophila melanogaster, as a model organism for his genetic research. Now, Mendel concluded that sex was irrelevant to the 7 traits he studied in pea plants, but Thomas Hunt Morgan realized that yes, sometimes the sex chromosome has something to do with the genes inherited. By the way, Thomas Hunt Morgan was the nephew of Confederate General John Hunt Morgan. Thomas Morgan also succeeded his uncle in making Kentuckians look good by winning the Nobel Prize in Medicine and Physiology for his work with sex chromosomes. Morgan's first goal was to identify the different phenotypes. He called the most common phenotype for each trait the wild type. In this case, for fruit flies, red eyes would be considered the wild type and white would be considered the mutant. And so the mutant is just a result of a mutation or a change in the gene from the wild type. When he mated a wild type female fly with a mutant male fly, all of the F1 progeny had red eyes, regardless if they were male or female. However, when Morgan did a reciprocal cross, the F1 females had red eyes, but the F1 males had white eyes in the offspring. These experiments suggest a relationship between the sex of the progeny and the inheritance of the eye color in fruit flies. Morgan proposed that the gene for white eye color in fruit flies is located on the X chromosome and that the Y chromosome does not carry an allele for this gene. So, females would have two X's, and the superscript capital E would indicate red eyes, and the lowercase e would be white eyes. And since males only have one X, all it takes is for one X to be affected by the recessive phenotype in order for it to express that particular phenotype. So in this case, the F1 generation was all going to be red-eyed. However, the females carried a lowercase e, in other words, a mutant eye color. And the next generation, all the females were red-eyed, either being homozygous dominant or heterozygous, and in the males only inherited the white-eyed allele. Morgan's hypothesis is called X-linked inheritance, or X-linkage. Females have two X's, which would then have two copies of the gene, and males would only have one copy of the gene. Since in fruit flies and humans, two X's make a biological female and an X and Y make a biological male, males get the short end of the stick with sex-linked disorders. That's because males only have one X chromosome, and if it carries a disorder, the male has it. Other examples include muscular dystrophy, red-green color blindness, as well as hemophilia, are all examples of human sex-linked disorders. The one I'm going to focus on is color blindness. So females can receive a sex-linked disorder if both of her X's carry the gene. However, she can be a carrier if her father is affected by a sex-linked disorder. So one great example of this is color blindness, red-green color blindness in humans. If you can't see a number in this diagram, you're affected by this disorder. Otherwise, you should be able to see a red-pink number 26 within a many dotted shades of green circle. So here are two scenarios. One where the father is colorblind, but the mother has normal vision and is not a carrier. So in this case, none of their children will inherit color blindness, but all of the daughters will be carriers since they received the lone colorblind X from their father. Potentially, these daughters could have colorblind children. In scenario 2, the father is colorblind and the mother is a carrier. Regardless of the sex, there is a 50-50 outcome. That's because the son could inherit the colorblind allele from his mother, and the daughter will definitely inherit the colorblind allele from her father and have a 50% chance of receiving the colorblind allele from her mother. So as you can see, sex-linked disorders can affect females. It's just much harder since both X chromosomes have to carry the allele. With the discovery of chromosomes, scientists have found that sometimes genes are inherited together if they are found on the same chromosome. Data on the percentage of recombinant offspring can be used to estimate the location of genes relative to one another on the same chromosome. Data on the frequency of crossing over can be used to create a genetic map, a diagram showing the relative positions of genes along a particular chromosome. The unit of distance between genes are called centimorgans, or map units. Morgan proposed that genes are more likely to cross over when they are far apart from each other than when they are close together. This explains why redheads have freckles and few do not. The genes for freckles and red hair are so close to each other on the same chromosome that they tend to be inherited together. So going back to Mendel's law of independent assortment, that only works if the genes are on separate chromosomes. If the genes are found on the same chromosome, it will appear as if the law of independent assortment has been violated, when in fact it hasn't. By now, we know that Mendel's rules do not apply in all circumstances. A strange type of genetics occurs in which the presence of one gene turns off the expression of another. This interaction between genes is called epistasis, which helps to explain why eye color is determined by 8 different genes providing a myriad of eye color types. Summer squash, which is mostly sold in yellow and green varieties, believe it or not, has a white variety. The color of the squash is controlled by 2 genes. As long as the dominant white squash gene, designated by capital W, is present, it doesn't matter whether the second gene, designated by Y, is dominant or recessive. The presence of the capital W outdoes, it turns off the effects of the Y gene. However, when the white gene is homozygous recessive, the genotype of the Y gene can determine if the squash is yellow or green. Let's do an example problem. Say that two squash plants are heterozygous for both genes. Since they're both contain a capital W, a dominant white gene, they're both going to be white in color regardless of the fact that the second one also contains a dominant allele. All right, when doing a dihybrid cross, you get the following genotypes. Again, the white gene is dominant to the yellow gene, so regardless of the yellow gene genotype, the presence of the white dominant allele will make the squash white. This is why 12 out of the 16, or 75% of the time, when you mate these two together, you will get a white squash. However, if the white gene is recessive, the yellow gene can now determine if the squash will be yellow or green. There's a 3/16 or 19% chance of a yellow squash provided that the yellow gene is dominant. If there isn't any dominant yellow gene and both of them are recessive, you will end up with a green squash, which is a 1/16 chance or 6.25% chance of it being green. So why is most squash yellow or green in gardens and grocery stores? Well, that's because gardeners and farmers breed only squash with recessive white alleles so that the squash turns out green or yellow. Though there is no difference in taste between a white or yellow or green squash, people like the colored varieties better. Mendel's findings are an important lesson in the evolving nature of science. While his ideas applied well to his pea plants and even many examples of genetics in plants and animals, they could not explain all areas of genetics. Without his work, we would not be able to understand genetic transmission as we do today. So other scientists came along, applied these principles in different situations, and found that there are other ways that genetics works. Now we know that genes are housed on chromosomes, and so our next chapter will look at the chromosomal effects of genetics.